Evening, folks. I hope everybody is doing as well as you can be tonight. This is going to be my first lecture in my series on gene expression and RNA. So if you are in my biology class, I recommend you go on the Blackboard site before you begin this, get a copy of the vocabulary sheets so that you can follow along, and also have some handy diagrams in there to illustrate concepts that I'm going to be explaining in the course of this lesson that are probably best represented if you have a visual to look at. I'm assuming that anybody listening to this has already heard my lessons on DNA. If you have not done that yet, I recommend you go back and listen to the DNA lessons first, because a lot of the concepts important for understanding DNA are going to be necessary for understanding RNA too. All right, so we know now how the DNA molecule works. In my last chapter, I told you all about how DNA is structured, how it encodes data in a series of A, T, C, and G nucleotides, and how it is replicated to make copies of itself in preparation for cell division. So it's great to know, okay, DNA stores all your biological information. It contains all the data on your hair color and eye color and length of your nose and all the other important basic details about you. But this does not answer the fundamental question of how do we get the data out of the DNA? We know how it's stored. How do we access it? Remember, let me just refresh everybody's memory about this, that what DNA really is a recipe book for proteins. Everything in your body is either made of a protein, built with the help of a protein enzyme, or regulated with the help of a regulation protein. So one way or another, proteins control all aspects of your structure and function. So by building all of the proteins needed to build you, that means that we can build or regulate all the parts of you and all the processes your cells carry out. So for our cells to do a thing, whether that thing is assembling a part needed for the body, whether that thing is carrying out a chemical reaction, we have to assemble the protein that is responsible for that structure or that process. Now the DNA fundamentally contains the recipes for all of those proteins, which we call genes. That's what a gene is, G-E-N-E. It is the set of instructions on how to assemble a specific polypeptide. Remember, a polypeptide is an amino acid chain. It may or may not be a complete protein. Some polypeptides are full, complete proteins by themselves. Some of them are just portions of a larger protein and will come together with other polypeptides at the quaternary level of protein structure to form something bigger and more complex. If you look at the individual gene, remember that the genes can be read in units of 3 nucleotides, which we call codons, C-O-D-O-N. And a codon, you might recall, is the specific formula for one amino acid. So having a line of codons in a gene is really giving us the instructions for the primary structure of that particular polypeptide. That is, which amino acids make it, and what order you have to put them in to assemble that polypeptide. So DNA encodes that data. We have a genetic language in which every 3 letters of the genetic alphabet is a word that represents a specific amino acid. We call that word a codon. DNA does not contain the amino acids. Let's not get confused here. DNA contains the instructions telling the cell, these are the amino acids you need to build this particular protein or protein parts. So what we need to do now is to get that data out of the DNA. We have to access that information and utilize it to make the protein we need. This is a process called gene expression. When you express a gene, that means that you are building whatever protein product that it codes for. If I'm expressing the gene for blue eyes, that means that my cells are assembling the protein that causes my eyes to be blue. So different genes are expressed at different times in our lives. There are genes that are expressed during puberty, for example, that were not active prior to that. There are genes that turn off when you're done growing. There are genes that turn on in a pregnant woman when she is gestating a baby that are not active any other time. So not every gene in your body is active at all times, and they are not always being expressed at the same level at all times. There can be varying degrees of activity of a particular gene depending on what's going on in your body. So one way or another, though, gene expression means decoding that information and building a protein according to those instructions. And that means the gene is being expressed. So in order to do that, The DNA contains that code. But you might want to think of the DNA as the master copy, the original copy of a big old rare book. That master copy is not the thing you lug out every time you need to quote from that book. If you want to quote from the United States Constitution, you do not have to go to Philadelphia and break open the case that the original document is stored in and drag out the Constitution if you want to read from it. No, what you have to do is, actually that's the wrong one anyway, the Declaration of Independence is in Philadelphia, Constitution's in the National Archives, my bad, sorry. But anyhow though, what you do in those cases is you make a copy, right? Rather than pulling out the original every single time you need to access the information in it, you make a copy of the data that you need. That, in a lot of ways, is how your DNA is going to work. We do not haul the DNA out of the nucleus every single time that our cells need to access some of the information within it. Rather, during the process of gene expression, what we are going to do is we are going to copy out a portion of the DNA. We are going to make a chemical copy of the gene that we want to express. That chemical copy will then leave the nucleus and go to a ribosome. The organelles, which you might recall from way back earlier in the course, synthesize proteins. And then the ribosome, using that genetic instruction, will build the protein that the cell needs. The DNA does not leave the nucleus during this process. It stays safe and intact inside of there. The copy of that information is expendable. If you break that one down, you can build a new one. So we keep the DNA in the nucleus. The DNA does not directly help building with all of the proteins that need to be built. Rather, we copy out the instructions from it. And it's those transcripts of the instructions that leave the cell. Those transcripts, by the way, are a molecule that we call RNA. RNA is a molecule that is very chemically similar to DNA, and it is essential to the process of gene expression. There is a basic rule in biology known as the Central Dogma, and that's a very important principle. We capitalize it, Central Dogma. The Central Dogma is a basic rule describing the flow of genetic information. It was devised by James Watson, one of the folks who worked on DNA, in 1957. And the Central Dogma of DNA basically talks about how genetic information moves around within the cell. And according to the Central Dogma, the flow of genetic information is that genetic instructions begin in DNA, are transcribed to RNA, and then are translated to make proteins. So our flow of information is DNA to RNA to protein. That is an almost universal rule in life. That's the normal flow you can expect every living thing to follow with genetic information. Starts in a DNA code, transcribe it to a molecule of RNA, and then use that RNA to translate it and make a protein. There are some weird exceptions. Most of them involve viruses. There are a few strange viruses that do this backwards, that they start with RNA and they use the RNA to build viral DNA. The HIV virus is an example of a virus that does that. But that is the exception rather than the rule. Normally you can expect DNA to RNA to protein. Now please understand that what I'm talking about here is information flow, data flow. I am not saying that DNA turns into RNA. I am not saying that RNA turns into a protein. I am saying information is in DNA. We copy that information to RNA, and then we read that information at the ribosome, which then builds a protein according to that set of instructions. Now, to carry this process out, obviously the intermediate step in here, between DNA, RNA, and functioning protein, the middle step of gene expression requires a molecule known as RNA. RNA is going to be our transcript molecule, if you will. It's going to be the intermediary between DNA and the ribosomes passing along the instructions. Now, RNA is very chemically similar to DNA. It is a nucleic acid like DNA is, but it's got several distinct differences from the DNA molecule. For starters, RNA is a single-stranded molecule. DNA, you know, is the double helix, the two strands wound around one another. RNA consists of only a single strand of nucleotides. Again, this is in almost all cases. There are a couple of weird examples of double-stranded RNA, but again, they primarily involve viruses. Secondly, we're going to find that RNA uses a slightly different chemical structure to build it. RNA nucleotides are built around a 5-carbon or pentose sugar known as ribose rather than deoxyribose. Remember, DNA is deoxyribonucleic acid because every DNA nucleotide is based around a little 5-carbon sugar molecule, a pentose sugar, that's what pentose means, 5-carbon, known as deoxyribose. RNA is ribonucleic acid, ribonucleic, ribonucleic. And that's because its nucleotides are made out of ribose, ribose. Ribose and deoxyribose are pretty chemically similar. Actually, the only difference between them is the placement of one hydroxide group attached to the molecule. But you will always find ribose in RNA. You will always find deoxyribose in DNA. Now, an RNA nucleotide has the same basic layout as a DNA nucleotide. It's going to have a 5-carbon sugar, a phosphate molecule attached to the 3' carbon, a hydroxide molecule attached to the 5' carbon, a hydroxide molecule attached to the 3' carbon, and a nitrogenous base attached to the 1' carbon. That basic structure is one you should already be familiar with from our DNA discussion. RNA and DNA nucleotides basically look the same. They even link to one another in the same fashion. They are built in the 5' to 3' direction, that you always link them together by joining the 5' phosphate of one nucleotide to the 3' hydroxide of the preceding nucleotide in the chain. So we always assemble RNA in the 5' to 3' direction, just like we do with DNA. Now, the other really big difference, however, comes with the nitrogenous bases found in RNA. You remember every nucleotide in DNA had one of 4 nitrogenous bases attached to it: adenine A, guanine G, cytosine C, or thymine T. Remember, those form the complementary base pairs between the 2 strands of DNA. A bonds to T, C bonds to G. You know what's in one strand, you can predict the other one. RNA also utilizes nucleotides that have a nitrogenous base attached to the 1' carbon. And in fact, most of those nitrogenous bases are the same. You will find adenine, guanine, and cytosine in RNA just like you do in DNA. However, you will never find thymine in DNA. There's never a T in a DNA sequence. If you're reading a genetic sequence and it's got a T in it, you know it's DNA, but RNA will never have that. Rather, RNA uses a different nucleobase called uracil, U-R-A-C-I-L, which we abbreviate with a capital U. Uracil is chemically very similar to thymine. In fact, uracil and thymine are so similar that uracil can form hydrogen bonds with adenine the same way that it can with thymine, or rather the same way that adenine can with thymine. So you can have an AU base pair just like you had an AT base pair. You will never find uracil as part of DNA. You will never find thymine in RNA. So if you're ever reading a genetic sequence and it's got a U in it, you know you're reading an RNA sequence, not a DNA sequence. So those are the most obvious differences between the 2 molecules in terms of their structure, that your RNA is going to have single-stranded structure. It's going to consist of ribose rather than deoxyribose sugars making up the backbone, and it's going to use uracil in place of thymine as one of the nitrogenous bases. You'll also find in general RNA is shorter, because RNA in general is storing a lot less information. DNA stores your entire genetic code. Your DNA molecule has your whole genome, or your DNA molecules, I should say, since they're split up among your chromosomes. But RNA is typically not going to do that, because RNA is not there to store all the data for you. RNA is there to move around specific units of data, genes, so that the cell can translate them. But once again, there are exceptions, and those exceptions, as you've probably guessed by now, are viruses. A lot of viruses utilize RNA rather than DNA as their source of genetic coding, including several viruses that you probably had during your lifetime. The common cold is an RNA-based virus. The flu is an RNA-based virus. And the one hopefully you don't have, COVID-19, the coronavirus that we're all concerned about right now, is an RNA-based virus. Actually, I should say the virus is CoV-2-19. That's the formal name of the virus. COVID-19 is the disease that it causes. But the coronavirus, like all coronaviruses, is RNA-based. But I digress. Now, RNA comes in 3 distinct forms. There are actually more than 3 forms of RNA, but But for purposes of our discussion, for understanding how your cells carry out genetic instructions and how they express genes, there are actually three distinct forms of RNA we're going to be dealing with: mRNA, rRNA, and tRNA. And you abbreviate those by putting a lowercase letter in front of the RNA part. So it's lowercase m, capital RNA, lowercase r, capital RNA, lowercase t, capital RNA, whenever you're writing that out. mRNA is messenger RNA. That's what it stands for. Your mRNA molecule is going to be a copy of one gene from the DNA, or as we put it, a transcript of that gene. The mRNA molecule is what we are making whenever the cell wants to express that gene. So that the DNA doesn't have to leave the nucleus and show the ribosomes what to do, we instead make a transcript of the desired gene. So the mRNA molecule is that transcript. It's the RNA copy of one gene from DNA that the cell wants to express. So we synthesize an mRNA molecule as the first part of gene expression. Once that's done, we are going to go out of the nucleus and take that to a ribosome. Remember, ribosomes, R-I-B-O-S-O-M-E, are the protein factories. They are the structures in every cell that build its proteins. There we will encounter rRNA, which is, as you might guess from the name, ribosomal RNA. Ribosomal RNA is only found in the ribosomes. Its entire job is to bind the mRNA. The ribosomal RNA binds to the mRNA molecule and holds it during the process of translation, where the ribosome is going to read that RNA molecule and see what it needs to do. And finally, we have tRNA. tRNA is transfer RNA, and what tRNA is transferring is amino acids. As the ribosome is reading the mRNA molecule to find out what it needs to do, remember the mRNA is telling it, how do I build this polypeptide? And a polypeptide is a chain of amino acids. So the transfer RNA is delivering amino acids to the ribosome. It is bringing them to the ribosome so that the ribosome can link them up and make the protein that that mRNA molecule is coding for. So mRNA is the transcript of the instructions. rRNA binds it to the ribosome. tRNA delivers the amino acids and puts them in place. If you think about it, it's kind of like, think like an old school factory, like where they used to manufacture things by hand in factories back in the day, that the mRNA is your work order. It's telling the factory, here's what you need to build, and here are the instructions to build it. Your rRNA is the working floor. It's going to take the instructions, receive them. That's the place where the work's going to be done. Your tRNAs are your workers. Your tRNAs are going to be carrying the parts and putting them where they need to go. All right, so that is our RNA. RNA, you can see, is doing a bunch of different jobs in the process of gene expression. So, how do we express a gene? A couple of steps are going to be involved here. You might have been noticing those steps if you were paying attention to what I was saying. There are actually two distinct processes we have to carry out in the process of gene expression to get from data stored in DNA to an actual functional protein that does a thing in your body. The first of those processes is called transcription. Transcription is how we go from DNA to RNA. In transcription, we are going to copy the gene from the DNA molecule that we want to express and make an mRNA molecule that is a copy of that gene. That mRNA molecule will then leave the DNA and go to a ribosome. The second step of the process is translation. Translation is when the mRNA molecule is read by the ribosome reads its instructions, and builds a polypeptide according to those instructions. So transcription makes the mRNA molecule, translation decodes it and builds the polypeptide that it codes for. And remember, that polypeptide may be a complete protein, or maybe it's just one smaller part of a bigger protein, and you need multiple polypeptides to put it together. I may use the terms polypeptide and protein synonymously throughout this discussion, but I'm really saying polypeptide. So, think back to our central dogma. What did I say? Genetic information begins in DNA, is transcribed to RNA, transcription, and then is translated to make proteins, translation. Those are our two steps that have to happen to express the gene. So what I want to do now is to take you through that story, to go through the process of gene expression step by step. So let's begin with transcription. The cell needs a protein. We don't care what protein it needs. That cell needs some protein to do an important job that it has to carry out. So first thing we have to do is to copy out the gene for that protein from the DNA. The process of transcription, if you are a prokaryote, is going to happen right there in the cytosol. If you're a prokaryote, everything happens in the cytosol because you don't have another space for stuff to happen anyway, right? If you are a eukaryote, this process happens in the nucleus. The nucleus is where the DNA lives. This is where RNA is going to be synthesized. Actually, you want to be more specific than that. We can be a bit more specific than that. Transcription happens at the nucleolus. 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 The nucleolus, you might remember way back in the day when we talked about the nucleus of the cell, that the nucleolus I described to you at the time is a region of the cell where RNA synthesis occurs. Right alongside the DNA, the nucleolus is the area in which RNA synthesis takes place. It's not a structure, it's not a compartment, it's just the area with its dark staining if you stain a microscope slide with a cell where intensive RNA synthesis is going on. So out in the cytosol for prokaryotes, at the nucleolus of the nucleus for eukaryotes. That's where transcription is going to happen. Now, transcription has a lot in common with DNA replication. A lot of the processes are the same. It's going to involve polymerase enzymes assembling a new RNA molecule. It's going to involve unzipping portions of the DNA. It's even going to involve moving in the 5' to 3' direction. A lot of the basic rules of DNA replication are also followed in transcription. In fact, the steps of transcription have the same names as the steps of DNA replication: initiation, elongation, and termination, in that order. But we're going to find that despite having similarities in names and similarities in some processes, that this process is very different, is going to give us a very different outcome. So let's start with step one. We're in transcription now. Our job is to make an RNA transcript of the gene that we want to express. So step one, initiation. Initiation is the prep phase. Now you might recall when we talked about DNA replication that I mentioned a little region of the DNA called the origin of replication, or ORI-C for short. And that the origin of replication was a non-coding region of the DNA whose entire purpose was to act as a binding site for all the enzymes needed for replication. Well, as it happens, you're going to find that you have that for RNA as well. That whenever it's time to make an RNA molecule, when it's time to carry out transcription, we're going to start from a place that does not code for anything. But there are lots of them in your DNA. You've got the one origin of replication, you work out towards either end. If you're a eukaryote, you might have a few of them. But for RNA, for transcription, at the beginning of every single gene, every gene in your DNA, there's going to be a little non-coding region at the start of that gene called the promoter. P-R-O-M-E-T-E-R, the promoter. The promoter, like the origin of replication, does not code for anything. It does not contain genetic information. It's a little stretch of DNA, about 10 to 100 nucleotides long, that acts as a binding site for the enzymes involved in transcription. That's its only function. So during initiation, what's going to happen is we are going to gather up all of the materials needed for this process at the promoter. The promoter is going to be the starting point for all of this. Primary thing we need to get at the promoter is going to be our polymerase enzyme. You might recall for DNA, we had polymerases that were the DNA building enzymes. And if you don't remember the spelling, that's P-O-L-Y-M-E-R-A-S-E. Polymerases are nucleic acid building enzymes. If you're a prokaryote, you only have one of these, and it is called RNA polymerase. Pretty straightforward. If you are a eukaryote, you have three separate ones. You have RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3, with Roman numerals 1 through 3, because you have one each for rRNA, mRNA, and tRNA. The one we are interested in building right now is mRNA. So RNA polymerase 2 is the one that is going to be required for transcription, because that's the one that builds mRNA. So during initiation, the enzymes that we need are going to gather at the promoter, RNA polymerase 2 if we're a eukaryote, just RNA polymerase if you're a prokaryote. We're also going to gather together a number of other essential support molecules called transcription factors, or TFs. The transcription factors are going to carry out miscellaneous support functions in the process of RNA replication, such as helping to unzip the DNA, helping to stabilize the DNA while we're doing the job, and a few other things. We're not going to go through all the different transcription factors involved in transcription. So we gather all these materials together. They all bind to the promoter. Once that has happened, we are going to begin to unzip the DNA. The RNA polymerase is going to begin breaking the hydrogen bonds between the two strands of the DNA. Now, we're not going to unzip the whole DNA like we do for DNA replication. We don't need that. We only need one gene. So we are only going to unzip the little portion of the DNA that contains that specific gene that we want to copy out. And you know what? We're not even going to unzip the whole thing at once. Initially, during initiation, we are going to unzip a little section of the gene about 12 nucleotides long. That little segment that's unzipped is going to kind of bulge outward from the rest of the DNA, and therefore we will call it the transcription bubble, because it's like a little bubble in the DNA. During the replication process, that transcription bubble is going to be where the action happens. This is where the ribosome is going to be, where it's going to be assembling RNA. So that is step one, initiation. We gathered together our RNA polymerases and our transcription factors at the promoter. They attached to the promoter, and we began unzipping the DNA to form the transcription bubble. Okay, initiation is done. Step two, elongation. Elongation is the building phase. This is the phase during which we are going to move along the length of the gene and assemble an mRNA copy of it. Now, here's the thing you need to understand. DNA has two strands, right? Remember this from our previous discussion, that the two strands of DNA are anti-parallel. They run in opposite directions. One of them has the 5' to 3' orientation, 5' at one end, 3' at the other. The other one is flipped upside down. It'll have the 3' at this end and the 5' at that end. So they run in opposite directions, hence anti-parallel. Remember, nucleic acids are always, always, always built in the 5' to 3' direction. This is the case with RNA as well. We are going to be building our mRNA molecule in the 5' to 3' direction. Now, because of anti-parallelism, that means that we're going to have to follow along the 3' to 5' strand of the DNA, because it has to be anti-parallel. So, in other words, to move 5 to 3, we're going to be doing so along the 3 to 5 strand of the DNA. So the 3' to 5' strand of the DNA is what the RNA polymerase is going to follow during elongation. It's going to move along that strand in the 5' to 3' direction. That is going to be our template for building the mRNA molecule. For that reason, the 3' to 5' strand of DNA, when we're talking about RNA transcription, is sometimes called the template strand. So the 3' to 5' strand of DNA is the template strand for building mRNA. It is sometimes also referred to as the negative strand or the antisense strand, A-N-T-I-S-E-N-S-E. And we'll talk about why that is momentarily. Now, the 5' to 3' strand of the DNA is not directly involved in RNA transcription. It is not going to be participating in the process. The RNA polymerase is not going to interact with it. It's there, but it's not doing anything. The 5' to 3' strand of the DNA, with reference to RNA transcription, is often called the coding strand, sometimes the positive or sense strand. And even though it's not directly involved in the process, you're going to see along the way that we call it the coding strand because it actually contains the code that we want to get it. All right, so how is this going to work? Now know the 3' to 5' strand of DNA is the template strand. The 5' to 3' is the coding strand. We have unzipped a little section of DNA that we call the transcription bubble. During elongation, the RNA polymerase is going to move along the template strand, always traveling in the 5' to 3' direction. As it does so, we are going to gradually unzip more nucleotides ahead of it. We're going to open up the transcription bubble ahead of the RNA polymerase. But the parts we are done with, we're going to close up behind it. So the bubble is going to re-zip closed behind the polymerase as we move along, and it's going to unzip open ahead of it as we move along. So what that's going to mean is that that little transcription bubble, that bulge in the DNA, is going to gradually move down its length along the way as we close up the parts we're done with behind ourselves and open up new parts ahead of ourselves. At any given time, there are going to be about a dozen nucleotides unzipped, close up behind it once we have copied them, open up ahead to allow more copying. Follow? So, RNA polymerase is going to move down the length of the template strand, traveling in the 5' to 3' direction. As it does so, it is going to assemble a new strand of mRNA alongside the template strand. We're going to build mRNA next to the template strand. We're going to build it by adding complementary bases to the template strand, the same as if we were building a DNA molecule. Now we're going to follow the same base pairing rules here. Chargaff's rules still hold up. A pairs with T, C pairs with G. So any place where there is a T in the DNA, my mRNA that we are building next to it is going to get an A. Any place where the template strand of DNA contains a G, the mRNA will get a C. Any place where it's a C, it will get a G. The only difference here from our standard rules is that any place that the DNA contains an A in the template strand, remember A normally pairs with T, but we never find thymine in RNA. We find uracil instead. So any place where the DNA contains an A, the mRNA we are building will contain a U rather than a T. So if my template strand has a codon that says CCA, then the mRNA will get a codon that says GGU. Make sense? So our polymerase gradually works its way down the length of the gene. Little by little, we are building an mRNA molecule alongside the DNA, following those complementary base pairing rules. Bonding to the template strand in the 5' to 3' direction. Because RNA is a single-stranded molecule, we do not need to worry about Okazaki fragments or going back and working backwards along the lagging strand or anything like that. We don't need to involve the coding strand at all. The template strand is the only strand we need for this process. You also might have noticed, if you're comparing this against our process of DNA replication, that I didn't mention anything about a primer. RNA can be built without a primer. DNA requires a primer to get started. RNA does not. And you might remember from our lesson on DNA that the DNA primer is actually a little tiny fragment of RNA, which obviously could be built without a primer of its own. So some of the things we involved with DNA replication don't need to happen here. We don't need to worry about Okazaki fragments and working backwards. We don't need to worry about DNA ligase to bind things together. We don't need to worry about a primer that needs to be built and then removed. So we just proceed unidirectionally. Remember, with DNA synthesis, it was bidirectionally. Origin of replication is in the middle of the DNA. We go outward in both directions, copying DNA towards either end. Transcription is unidirectional. We always flow in the same direction. We start at the beginning of the gene, that's where the promoter is, and we work 5 to 3 down to the end of the gene. So proceeding in a single direction rather than bidirectionally, like DNA. Now, we are assembling this new DNA sequence, but here's an important thing about this. The very beginning of the molecule, when you were not a DNA sequence, an RNA sequence, my bad. When you're building this RNA, the very first codon of the mRNA molecule is going to be the same for every mRNA. The first codon assembled during transcription will always be AUG. AUG. That is always the first segment of any mRNA molecule. And therefore, if you looked at the template strand of the DNA, that means that the first codon in the template of every single gene is TAC, because that comes out to AUG in the RNA. Why is that? The reason is because the codon AUG has a special role in this process. You might recall we talked about codons, that I told you that there were 60 possible codons, the 64 possible codons for all of the 20 amino acids. Well, that's way more than we need to code for 20 amino acids. I told you at the time that most amino acids had redundancy in their genetic coding, that there was more than one codon that represented any given amino acid, sometimes as many as 6 different codons representing the same amino acid. Well, I also mentioned at the time that there were a few codons that had other jobs. AUG is one of them. AUG is the start codon. Every single mRNA molecule begins with that one because AUG is the signal that tells the ribosome during translation, here is where you begin. That lets the ribosome know this is the spot that you start reading from. So every gene has to have a start codon at the beginning so that the ribosome knows where to start. The start codon actually does code for an amino acid called methionine, and therefore methionine is always the first amino acid in our polypeptide. All right, sounding good. So, little by little during elongation, we work our way down the template strand 5' to 3'. We assemble mRNA alongside the template strand, following Chargaff's rules of complementary base pairing, except that wherever the DNA would normally get a T, the RNA gets a U instead. One last thing I wanted to show you here, and I have to sketch this out so that you can see what I am talking about. Bear with me one moment. Okay, so check this out. What have we got? You can see I've got a little DNA sequence. Obviously, I only drew a few letters just to show you what's going on. 5' to 3' strand of the DNA is the coding strand. 3' to 5' strand of the DNA is the template strand. And so we've got a sequence here that goes AACTGA in the coding strand. And therefore, the template strand is going to say TTGACT. Okay? Now, remember, we're building a new molecule of mRNA alongside the template strand, but we have to follow Chargaff's rules. Wherever there's a T in the DNA, you get an A in the RNA. Wherever there's a G in the RNA, a G in the DNA, you get a C in the RNA. Only difference is any place where the DNA has an A, you put a U instead. Now check this out. I'm going to draw in the mRNA that we're going to synthesize here. Following Chargaff's rules, I put in an A beside every T in the template strand. I put in a C beside every G. I put in a G beside every C. And beside every A, I put a U. So here is the mRNA that I just built. AACUGA. Got it? Now I want you to notice the thing. The mRNA that I just built, first codon of it, AAC. Look at the coding strand of the DNA, AAC. Second codon, UGA. Coding strand of the DNA says TGA. Same except thymine instead of uracil. In other words, folks, the reason we call the coding strand is because the coding strand contains the code we're actually trying to access. That is the actual code for the gene that we want to copy. Hence why we call it the positive or sense strand. The template strand is the negative or anti-sense because it's the opposite of the code that we want to copy. But because A has to pair with T and G has to pair with C, I can't use the coding strand to build my RNA because A won't go with A and C won't go with C and so forth. I need to use its opposite. So you can see that even though the coding strand is completely uninvolved in the process of transcription, the sequence we end up building will look like the coding strand when we are done. Only difference will be that it will contain U's instead of T's. Does that make sense? Just hold that there for a minute so you can all copy those letters down. This was just a random sequence that I made up on the fly in order to show you how things work. Okay, so that is elongation. We work our way little by little down the length of the gene. At last, we get to final step, termination. Now, termination here is going to proceed a little bit like what we saw with DNA. Remember that in DNA, when you get to the end of the mRNA, whenever you get to the end of the DNA molecule, you find a termination site that it basically tells the replosome, hey, you're done making new DNA. We have something similar with RNA transcription. At the end of every gene, you are going to have a terminator. And the terminator is going to basically tell, it's going to be a non-coding sequence of the DNA that tells the RNA polymerases, hey, you're done. So we keep building new mRNA until we hit the terminator. And that signals that we are finished. At that time, we stop building new mRNA. The mRNA molecule that we have been assembling this whole time detaches from the template strand, and the transcription bubble closes up. What we have now got, folks, is an mRNA copy of one gene from the DNA, copying it from beginning to end. The first codon of that mRNA is always the start codon, AUG. At the end of it, you will always find a stop codon as well. The stop codon is basically there to tell the ribosome in the next step, hey, you're done. You have finished copying everything you need to copy. You're finished. We've built the protein. So you have a start codon at one end of the mRNA that tells the ribosome, here's where you begin reading, and a stop codon at the far end that tells the ribosome, stop building now. You have finished making the protein. There are three possible stop codons, and I actually had one of them in my little diagram that I built here. UGA. UGA is a stop codon. UAA is also a stop codon, and so is UAG. So any of those three sequences, UAG, UGA, or UAA, all three of those have the same genetic function. All of them are stop codons. Every time we build an mRNA molecule, one of those three will be the last codon in the sequence, because that basically is going to tell the ribosome, here's where we are done. All right, so what we now have is an mRNA molecule that is a transcript of the gene that we want to build. Sometimes we just call it the transcript. Now, a term that you're going to see coming up a lot in your biological literature. A lot of times, the molecule that we just assembled at this time is called pre-mRNA. A lot of times, they'll say that what you're building in transcription is pre-mRNA rather than mRNA. Because before it can become complete mRNA, we have to do one more thing to it. Before we can send it on for translation, we're going to have to carry out a process called splicing. But you know what? I'm going to tell you about splicing after we talk about translation, because honestly, knowing some things about translation will be helpful to you in understanding why splicing matters. But we have finished our process of transcription. We have just completed making an mRNA copy of the gene that we want to express, or a pre-mRNA transcript, if you will. After the splicing process is completed, our mRNA, which is now completely an mRNA after splicing, is going to leave the nucleus. It's going to exit through one of the nuclear pores. Remember those from when we talked about the nucleus before? P-O-R-E-S. So it's going to go out through one of the nuclear pores, and from there it's going to travel to a ribosome. The ribosomes, you might recall, are the protein factories of the cell. They are the structures that are responsible for building all of your body's polypeptides. And that's what's going to happen next, the process of translation, in which the ribosome is going to decode those mRNA instructions and assemble the polypeptide that the cell wants. Incidentally, I want to point out a little thing about this. You also might remember when I told you about ribosomes before. I told you that some ribosomes are free-floating in the cytosol of the cell, whereas other ribosomes are bound to the endoplasmic reticulum, forming rough ER, as we call it. And there is a difference in which ribosomes do which jobs. The general rule, and this is not a hard and fast rule, but for most proteins in our bodies, is that if a protein is going to stay inside the cell and do things within the cell itself, that protein is going to be made by a ribosome attached to the rough ER. Sorry, to the smooth ER, my bad, smooth ER. So any protein that's staying within the cell is going to be made by No, what am I talking about? Smooth ER doesn't have ribosomes. Okay, back up. Any protein that is going to be staying completely inside of the cell and doing things for that cell is going to be made by free-floating ribosomes, by free ribosomes that are not attached to the endoplasmic reticulum and that are just drifting around in the cytoplasm. On the other hand, if the protein we are making is either going to become part of the cell's membrane, or if it is a protein that is going to be exported from the cell to someplace else in the body, then in those cases, the ribosome that builds it will be attached to the rough ER. In other words, ribosomes on the rough ER typically build proteins for external use, proteins that are going to be attached to the membrane of the cell or are going to go someplace else in the body. Free-floating ribosomes typically build proteins that are going to stay within the cell and do things for that cell. This is not a hard and fast rule, but it's a general rule of thumb. And that's usually where you're going to find your ribosomes dividing up their labor, and therefore that's going to determine where the mRNA molecule goes. All right, so that's transcription. That's the first half of our gene expression. I think I've given you enough to chew on right now. This is about a complete lesson, so I think I'm going to call it a day. For our next part, we're going to have a two-part lesson on RNA. We're going to talk about the process of translation and find out how we go about decoding these instructions to build our polypeptide. And then we're going to talk about that thing called splicing, find out what we have to do to the mRNA before it can be translated, and we're going to find out why that matters for the question of how many genes you actually have, which is a really interesting and surprising thing that we learned fairly recently. All right, thank you all for your attention. I hope you all have a great night. Oh, my camera didn't shut off. Sorry. Okay, hope you all have a great night.